Hi guys, in this video we are going to learn about glycolysis. So let's give ourselves in the overview of glycolysis. It is a very important biological pathway which occurs in every life form as we know it. So first of all, we need to know that all biological organisms require energy to survive. So glycolysis pathway is one of the pathway which cells use to transform sugars into energy in the form of ATP. So the overall process is happening just to produce the ATP molecules so that the cells can use the ATP to do the basic functions. So the whole process. So what is the whole process, the pathway? It is described as the oxidation of glucose pyruvate with a generation of ATP and NADH. This whole process produces ATP and along with other products like NADH, which can be later used to produce even more ATPs. Okay, so the glycolytic pathway describes the oxidation of glucose. So, how the oxidation is happening? There are lots of enzymes are involved in this. There are total 10 enzymes which are involved in this whole process, which we will be discussing in more detail. So, glucose to pyruvate with the generation of ATP and NADH. The whole process is basically happening just to produce more and more ATPs. Glycolytic breakdown of glucose is the sole source of metabolic energy in some mammalian tissue and cells. Every cell needs glucose, like for example, the glucose is the sole source. Glucose, a sugar, is the sole source which will be break down into pyruvate molecule. And while this breakdown, the ATP molecules will be produced. Glycolysis is a universal pathway which is happening in all organisms, from yeast to mammals. Anaerobic microorganisms are entirely dependent on glycolysis. As the glycolysis process is happening, there is no need of oxygen. So anaerobic microorganisms, they are entirely dependent on glycolysis only. It is also known as Embden-Meyerhof pathway and it occurs in cytoplasm. So cytoplasm is the location where the whole glucose is breaking down into two pyruvate molecules. So let's see the reaction. For example, the whole process is occurring in 10 steps. So from glucose to pyruvate. Okay. This whole process is happening in total 10 steps. So these 10 steps are divided into two phases. One is energy investment phase in which 2 ATP forms 2 ATP and occurs from 1 to 5. You don't need to, if you are not getting this point, we will be discussing it in detail. You just have to remember that this whole process has been divided into energy investment phase, energy payoff phase. And how this whole energy investment phase, energy payoff phase, what are all these? We will be discussing it in further detail. So let's see this diagram. So for example, this glucose is here, the sugar which is present here. Okay. So first 5 steps, this is just the overview. I'm just explaining this that from glucose, when glucose, more steps are involved here, and the glucose, glyceraldehyde 3 phosphate and dihydroxyacetone phosphate, which is also known as DHAP. When glucose and glyceraldehyde 3 phosphate and dihydroxyacetone phosphate, these are produced. So this is known as energy investments phase. Why? Because here we are providing, yeah, we can say that we are giving ATP as an input energy. We are investing ATP here so that the whole process could be completed. But later, what happens after that, during when glyceraldehyde 3 phosphate further goes, this process happens and dihydroxyacetone phosphate also produces pyruvate. So during that process, from 5 to 10 steps, there here the ATP output is more than the input. Okay, so let's understand that in detail. So this is energy payoff phase and this is energy investment phase. Let's discuss it in detail. So first is preparatory stage where the preparations will be occurring, like energy investment phase. So this step 1 is known as phosphorylation of glucose. All you need to know is that the glucose, here is the structure of glucose, simple glucose. And as I said, that this is the energy investment phase. So we are giving ATP as an input so that our glucose could be transferred into glucose 6 phosphate. So ATP has been provided here. Glucose is producing, or we can say that glucose molecule to form glucose 6 phosphate. This is the first step, and this whole process has been possible because the enzyme hexokinase here was present. So hexokinase enzyme phosphorylates the glucose molecule to form glucose 6 phosphate. So first reaction is glucose produces, forms glucose 6 phosphate with the help of hexokinase. Hexokinase, and the one ATP molecule has been used here. Okay, so this is why this is called energy investment phase. In the step 1, we are producing ATP molecule so that our glucose could be formed into glucose 6 phosphate. And enzyme hexokinase is used here. Second reaction is isomerization of glucose 6 phosphate fructose 6 phosphate. So, from glucose to glucose 6 phosphate and from glucose 6 phosphate to fructose 6 phosphate. So, this whole glucose has been formed into fructose 6 phosphate, and here the enzyme phosphohexose isomerase has been used. Okay, this is the second step. Third step is phosphorylation of fructose 6 phosphate. So, first say again, glucose to glucose 6 phosphate, 6 phosphate, glucose 6 phosphate, from glucose 6 phosphate to fructose 6 phosphate, and from fructose 6 phosphate, we are producing fructose 1,6 biphosphate or bisphosphate. Here we are again, ATP has been provided to fructose 6 phosphate so that it could be transformed into fructose 1,6 bisphosphate, and the enzyme phosphofructokinase 1 has been used here, and which is an important enzyme in terms of regulation. So as we have seen in step 1 also, ATP was provided. Step 3 also, ATP has been provided here. So fructose 6 phosphate has been transformed into fructose 1,6 biphosphate. Step 4 is cleavage of fructose 1,6 biphosphate. Okay, so here what happens is this fructose 1,6 biphosphate, with the help of aldolase, it splits the 6 carbon sugar phosphate to form into 3 carbon sugar phosphate. Okay, so one is dihydroxyacetone phosphate, which is also known as DHAP, and one is glyceraldehyde 3 phosphate. They both are 3 carbon compounds. Fructose 1,6 bisphosphate helps split the 6 carbon sugar phosphate to form the 3 carbon sugar phosphates, and they both DHAP will also produce a pyruvate. Okay, and this glyceraldehyde 3 phosphate will also provide the pyruvate because they both can be like, it can change into glyceraldehyde 3 phosphate too. So they both are reversible. Okay. So this was the step 4. Now, step 5 is conversion. As I said, that they both are convertible. Like dihydroxyacetone phosphate can be converted to glyceraldehyde 3 phosphate. So, DHAP can be converted into diglyceraldehyde 3 phosphate. Okay, with the help of triose phosphate isomerase. So, once the DHAP has been converted into glyceraldehyde 3-phosphate, we have 2 glyceraldehyde 3-phosphate. Now, both glyceraldehyde 3-phosphate will undergo the process and they both will produce 2 pyruvate. So, once again, the DHAP will be converted into 2 glyceraldehyde 3-phosphate, and these 2 glyceraldehyde 3-phosphate will undergo the whole process and they will produce 2 pyruvates. One pyruvate here, one pyruvate here. So this was the whole preparatory stage or the energy investment phase where we provided ATP. Okay. Now the payoff phase begins. So this is all the harvestry phase, which is also known as the payoff phase. Here, the oxidation of glyceraldehyde 3-phosphate is occurring with the help of dehydrogenase. Here we are using NAD also, okay, which glyceraldehyde 3-phosphate and NAD. This whole dehydrogenase enzyme, it catalyzes the oxidation of aldehyde group to a carboxylic group, okay, which also phosphorylates. This glyceraldehyde 3 phosphate will be phosphorylates and it becomes 1,3 bis phosphoglycerate, okay, and one NADH is produced here. This NADH can form 3 ATPs later. Like you can use that in another processes also, like this can be used in electron transport chain too, and here there it will produce more ATPs. Okay, so this is the payoff phase where we are getting ATPs. Next is transfer of phosphoryl group from 1,3 phosphoglycerate to ADP. So what happens here is, here we are also again getting ATP. Okay, so 1,3 bis phosphoglycerate, here we are using kinase. Okay, so what happens here, that phosphoglycerate kinase 1,3 bisphosphoglycerate, it loses a phosphate group which is transferred to ADP molecule, which forms the ATP. Okay, so it loses one phosphate, so it becomes only 3 phosphoglycerate, and ADP on by gaining one phosphate molecule, it becomes ATP. So again we are getting more energy, more ATPs. In the previous also step, we got NADH, which was used to produce ATP, and in this process also we are getting more ATPs. Okay, so here what happens, 1,3 bisphosphoglycerates, it gets, with the help of kinase, it loses its phosphate group which is transferred to the ADP and it becomes ATP. And by losing one phosphate, it becomes 3 phosphoglycerate. Okay? All you need to remember that we are doing this whole process twice. Why? Because we have 2 glyceraldehyde 3 phosphates. Okay? 2 molecules of glyceraldehyde 3 phosphates are present here. We are just showing the one molecule. We are just showing the whole process once only. But these are occurring twice. So we are getting twice amount of NADPH and 2 ATPs. Like 2 NADH from the previous reaction and 2 ATPs here if we get the 2 glyceraldehyde. Okay. Now the next reaction is conversion of 3 phosphoglycerate to 2 phosphoglycerate. Again, one phosphate, mutase is an enzyme here. Okay. So phosphate group on carbon 3 is removed and the one on carbon 2 remains. So the 3 phosphoglycerate becomes 2 phosphoglycerate. All you need to know this. But 3 becomes 2 phosphoglycerate here with the help of mutase. Now, in the ninth reaction, it is a ninth step where dehydration of 2-phosphoglycerate, where the removal of H2O from 2-phosphoglycerate occurs. Dehydrogenation occurs where we get H2O. Okay, so where here we get phosphoenol pyruvate. So enolase is helping in dehydrogenation. So 2-phosphoglycerate, with the help of enolase, it becomes phosphoenol pyruvate and one hydrogen molecule we get here. And in the reaction, in last reaction, step 10 is transfer of phosphoryl group from PEP to ADP. So this phosphoenol pyruvate, okay, and here we are ADP is given here. Okay? Okay. So pyruvate kinase forms ATP when converting PEP to pyruvic acid. So when the whole phosphoenol pyruvate is being transferred to pyruvate, this ADP gets the phosphate molecules and it gets transformed into ATP. So here again we are getting two ATPs. Okay. So the overall reaction, when this whole reaction was occurring, one ATP was used in the step 1. One ATP was used in the step 3. But after the payoff phase, we got 2 glyceraldehyde 3 phosphate, and after that we got 2 NADH and 2 ATPs. And here also in the 10th step, we are getting 2 ATPs. Okay. So this is the total ATPs we are getting here. 2 NADH and 4 ATPs. But if we minus these 2 ATPs from here, we are getting 2 NADH and 2 ATPs. I hope this whole reaction is making sense to you. Okay. So thank you so much for watching this video.